What does a mouse in a cage, a lion in the wild, and a person at a mall all have in common? Today's guest has built a scientific career exploring that question, not just through observation, but by uncovering how the brain itself composes meaningful behavior in a complex world. Dr. Sandeep Bob Datta is a professor of neurobiology at Harvard Medical School and a leading voice in what might be called next-generation ethology. His lab asks a deceptively simple question: how does the brain enable animals to move through the world with purpose? That is, how does sensation inform action, and how does action in turn shape perception? Bob is best known for pioneering research on olfaction, how animals, especially mice, use their sense of smell to explore, survive, and learn. But more broadly, his work blends neuroscience, behavior, and machine learning to decode how natural, spontaneous movement arises in real-world environments, where animals like us act on their own desires, not in response to a lab bell. His lab's innovations include motion sequencing, or MoSeq, a method to break down complex behavior into its sub-second building blocks, sort of like recording the grammar of body language, using tools like functional imaging, optogenetics, and custom machine learning models. Bob is rewriting how we think about behavior, not as a reaction, but as a structured expression of cognition. Today, we talk about brains, behavior, and an unexpected path to therapeutically relevant discoveries. We will also talk a little bit about kind of today's scientific and funding environment that enables all of this work and areas where it might be threatened. Bob, welcome to Let's Talk About That.
Oh, thanks so much, Anirvan. I'm glad to be here.
Well, we have known each other for a while and have followed your work with fascination. And especially, you come from a pretty deep molecular background. The work that you were doing in Mike Greenberg's lab, Richard Axel's lab, et cetera. And then when you started your own lab at Harvard, I kind of noticed that you had gone in a direction that is a somewhat unusual direction for molecular biologists, right? Molecular biologists pride themselves on a highly reductionist view of the world. They count their achievement in genes cloned or understanding the function of domains of proteins. And you kind of broadened your lens from that perspective. Somehow you decided that what you want to study is almost at the opposite end of the spectrum, and which to many cell and molecular biologists would seem very complex, which is the area of behavior. So tell me a little bit about what drove you to study behavior, or to even think that it was a tractable problem.
So that's a great question. So it turns out that unsurprisingly, when we started working on the brain and behavior, really our intention was to think about it from a kind of molecular biology perspective. So, we think of genes as talking to each other and building kind of nice linear pathways that enables cells to interact usefully with signals in the world and to adapt. And, you can think of animals and animal brains as kind of doing the same thing, right? And so the initial question we wanted to address in the lab was deceptively simple. We wanted to understand, for example, why mice run away from the smell of a cat. And so if you're thinking about how that works, right, you imagine a pretty linear system, just like in molecular biology, where there'll be some cat smell detector in your nose, and that detector will be hooked up to some center in your brain that mediates escape, and some trigger will be, flipped, and then the mouse will escape. And so when we started the lab, really my interest was in trying to figure out these kinds of simple reflexive behaviors and to think about them from a kind of traditional, linear, very comfortable from molecular biologists and cell biologists perspective.
Got it. Okay, so let's talk about reflexive behavior, because I think what is fascinating and great about behavior is it's something we can all relate to, and we do all the time. Like that is what we are constantly doing. We're behaving, right? And reflexive behavior, instinctive behavior is interesting because things like fear. You talked a little bit about fear. They have a certain innate thing. My son hates snakes. I'm assuming that whatever tens of thousands of years of human evolution have like encoded that into his brain, that is not an animal to be petted. And there are, of course, complex behavior between that. So, there's the behavior between us and an animal, like the one we talked about. I'm fascinated by dog behavior. I have a dog, and the dog owners in my life, they just love dogs, frankly, because of their behavior. Like,
Yeah.
because dogs have a way of connecting with people that no other animal does. Um, and then behavior between people has a very strong interaction element, right? You are evaluating everything. You're talking to someone, like, are they bored? Are they interested?
Right.
You're out on a date. You're thinking, like, based on that small interaction, you're processing whether you need to cut it short or whether you're trying to go on the next date. So behavior is all over, and there's a lot of, like, computation behind it. But it also seems incredibly complex, right? Which is
Right.
why we cannot predict it. Like,
Right.
you interact with another person, you can say, well, I don't know how they're going to react, and so complex behavior has its own thing, and even for even simple behavior is uh, quite complex in its own way. And what I found fascinating is that you started thinking about how to break down behavior in part by looking or analyzing the motor output carefully, because that's a way of thinking about it, right? Because when you say
Mm,
behavior, it has a lot of valence associated often with it, right?
Correct. That's
And
right.
that's how you think about it. But if you say, like, pretty much anything that you know about behavior is the motor output of another person or animal, and uh, speaking as you're doing now is a
Yeah.
very common and richly developed human behavior. Um, so tell me a little bit about your thinking that about breaking down behavior
Sure.
into elements that one could study, and how do you decide to approach that?
Yeah. So, initially when we decided to study behavior, to be honest, our goals were very simple and entirely by accident we end up in this space. So, like I was telling you before, we're interested in, for example, why is it that a mouse runs away from a cat? And actually, the first thing that a mouse will do when you give it any odor, let's say a food odor or a predator odor, is of course sniff, right? And so if you're interested in understanding, how the brain enables animals to respond to smells, the first thing you need in the lab is a sniff detector. That's the very first thing you need, right? Like, how do I know the animal is responding to the smell at all? And there's lots of ways you can detect sniffs in the lab. And we were thinking about different ways we could do that non-invasively, where we didn't have to implant something into the animal's nose to measure airflow or something.
Right.
And around the time we were thinking about this problem, a student in my lab was really into video games and was playing with a gaming platform that was popular back then called the Microsoft Kinect. And I don't know whether you remember the Kinect, but the Kinect was a gaming platform where, there was a 3D camera that you would dance in front of, and you could play tennis or you could
Oh,
play.
yes,
Remember
yeah,
that?
yeah.
Remember that?
Kind of like was it like Wii or something? Was it
Yeah,
like
it's like a Wii, but they like your body was being imaged, and so you could kind of interact. You could move your
got it?
body around and interact with the game,
Yes.
right?
Yeah.
And so there was a sensor, right, that was seeing you in 3D. And so this student of mine, Alex, literally went up the block with $100 and bought one of these sensors, these 3D sensors. And instead of pointing it at me or the other graduate students, we pointed it down at the mouse. And the idea would be that when mice sniff, they rear up on their hind legs, right? And so if we could use this 3D sensor to just detect whenever that one thing was happening, we would kind of have invented a sniff detector, and that would teach us a lot about how smell works. So we went ahead and built this thing. Like, we put a mouse in a bucket with one of these 3D cameras on top of it, and we let a mouse run around. And when we looked at the data, what we discovered was that the data were really chunky or blocky. That is, the mouse seemed to do one thing in 3D, pause for just a millisecond, and then switch to doing another thing in 3D, pause for just a millisecond, and then switch to doing another thing. So it looked like its behavior was, when you looked at it in 3D through this camera, was actually organized out of parts, out of little three-dimensional stereotyped movements. And when we looked at them, there were things like, the mouse rearing up, or the mouse rearing down, or the mouse turning right, or taking a sniff to the left, or turning its body to the right. And so just by looking at these 3D videos, we could recognize that the animal seemed to be organizing his behavior into these modules or parts. And then we were hooked. Then we realized that we had something that was much bigger than just a sniff detector, we get a way of maybe determining what the different parts of behavior are that a mouse is expressing in a given circumstance.
Got it. Uh, so it's interesting. So you're saying that, because I think I've often thought of behavior as like a continuum. Like you walk into that, like you just think that's what it is, right? Because it seems very analog in some way, right? That you're just doing something, and it's you're never really doing the same thing twice, right? And no
Right.
two conversations are the same.
Right.
Even if you're walking to the grocery store, which I've done 100 times, it's a little bit different each time. And at a macro level, I remember, reading things about stereotypical behavior, which is if you just were to ask, let's an animal exploring an environment, it would do it in some pattern. Whatever goes to the corners first and then to the sides. Or there's a fascinating study following people because you could track cell phone movement about how stereotypical our behavior was in the scale of weeks or something. Like you'd say, like we think we do a lot of different things every day, but if you follow your phone, it has a track that it is following.
Yeah,
And
that Barabasi study is terrifying, right? Because basically,
yeah.
we're entirely predictable. Like there
Yes.
were
Okay. Right. So yeah. So I'm not imagining the study,
yeah, no,
right?
no, 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 no. That's a terrifying
Okay.
study.
So that study. So
Yeah.
so that study was at a very different time scale, right? So then you say, like, okay, if you look at a large time scales, animals do this, and that I'll say everyone kind of relates to it. Like as anyone with a pet or, like I said, I have a little dog. When he comes home, and he has a certain sequence. He wants to play for a while, and so he wants me to chase him around, and I chase him around. Then he gets tired and he rests for a bit, and then he wants to eat. And he kind of does that every day. He doesn't get bored. And then he gets quite upset if it's broken, by the way. So like
Right.
he'll start like pawing me, saying, "Okay, like dude, it's time to kind of play." And so at a macro level, I can see, yeah, it's pretty stereotypical. However, the fact that if you look in a more refined time scale, you find these little highly stereotypical bits of behavior was something I think no one would have expected, and I was certainly surprised to hear that. And so, tell me a little bit about this. Like, I guess at what time scale are these little bits being put together? So, how long are these little segments that you notice? And I'm assuming you and your student are just watching things
Yeah.
on your screen or something, and you start noticing these little chunks of behavior. Is that right?
That's exactly right. And then when you kind of zoom in time, what you discover is that these chunks are organized at the sub-second time scale.
So,
So
okay, very, very short
very
bits, if you will.
fast. Yeah. So like on average, each one of these little elements of behavior in a mouse lasts 400, 500 milliseconds. So not very long.
Right.
Perhaps even under the level of perception of humans, which I think is a very important point. When we think about behavior, we bring
Yeah.
our own limitations to the analysis, right? We want to gain some intuition with our eyes. And
Yes.
it really was only by essentially slowing down the videotape that we could really convince ourselves that there are parts that are organized, at this relatively fast time
But
scale.
very, very short time scale, less
Yeah,
than a
yeah,
second, right?
yeah. So,
And is it, do you expect, or is it known whether our behaviors follow similar kinds of algorithms and have similar bits of sub-second behavior that all patch together to create something that looks smooth?
yeah, so we believe that at some level that's going to be true. And we know that a little bit because we've applied some of the machine learning methods that we've developed for animals to humans. And so we can see in certain data types, like accelerometer data that you have in your wrist, we can definitely see kind of the organization of behavior. Now, the time scales of that are going to vary, right, depending on the animal and the context in which they're behaving. I will mention, though, that this sub-second time scale that we're describing
Yeah.
does have analogs in humans. So the 300, 400, 500 millisecond
Mm-hmm.
time scale is about the time scale at which your eyes saccade. So when you're looking in a
Ah,
room
okay.
and you're kind of using your eyes to explore, that's the fundamental time scale at which that behavior is organized.
Oh, it's very interesting. So, when talking about eye movement, so yesterday I was watching Stephen Colbert, and Joaquin Phoenix was his guest, and he's a bit of an awkward guest. He really doesn't like talking very much, and Stephen Colbert was struggling to kind of get two words out of him. Brilliant actor, and so he said like something along like, what do you want to do? Like Stephen Colbert's like struggling a bit, and he says like, can we have like a minute of awkward silence? Okay. And so they proceed to do that, and the camera is, smack on their faces,
Yeah.
and then I'm watching that awkward silence play out, and it is so much of it had to do with the eyes, right? So you see Walking Phoenix's eyes, like, like just moving a little bit, and awkward silence has a very significant eye component, and so if you can, if you go look at that video, you
Yeah.
should see it is probably moving at that 400 millisecond time scale.
Ah,
Um, and it also gets to this thing that, when people talk about you know, we read a lot into behavior. Like people will often talk about it. Like human to human is very rich in sensing behavior, right? And often it's hard to describe it in words. We often don't know exactly what is happening. If
that's right.
somebody makes you feel
Right.
uneasy or comfortable, or they seem a bit angry, but maybe we have innately also developed the ability to kind of detect those things at very short time scales and interpret them,
Yeah.
even if we cannot give it a very specific name, right?
Right. So, we're used to thinking of ourselves as having free will and being in total control of everything that we do. And in doing so, we think of ourselves as being fundamentally unpredictable. Like we don't think of ourselves as
Yes.
predictable creatures, right? But the very notion of body language, the idea that I can look at you and you can look at me and we can communicate at a distance without words using just our bodies fundamentally means that our behavior has to be predictable, because what body language is really, right, is me predicting your emotional state or what you're thinking about based upon movements that you're generating, right? And so I'm literally making
Right.
predictions about you based upon your movements, and you're doing the exact same thing to me. And the fact that we can make predictions about each other
Yeah.
based on behavior means that behavior must have a lot of structure to it, right? And that
Yes.
structure is kind of what we're unveiling with these AI machines. Like we're unveiling the basis of body language. It's because it's predictable that you can understand it.
Right. Yeah. Yeah. No, it's fascinating. So getting back to your mouse experiments, and you've had a deep interest in olfaction, and that's even how this project got started. And I know you continue to work on olfaction quite a bit. And I want to kind of follow the thread about how the work starting there more recently has created some really interesting insight into Alzheimer's disease, which I think people would not think of you as being an Alzheimer's researcher. So just to kind of preview it for our audience, that I think it's a fascinating journey from how one goes from very fundamental observations, and you could end up in something that is potentially therapeutically important. But getting to the early points and your interest in olfaction and the path that eventually led to that. So, uh, and that itself is interesting. So maybe we can spend a
Sure.
minute talking about olfaction and a little bit of what is known or not, because you worked in a lab that trained in the Apsil lab that kind of discovered the olfactory receptors,
You said?
and I'd love to hear a little bit about how that part is organized. But you talk about parts of the olfactory system that were not fully understood that
That's right.
allowed you guys to discover a new class of receptors that might
That's right.
serve a function for a complementary sense
That's right.
of smell. So tell us a bit about
Yeah,
that.
yeah. I mean, so smell is a mystery, right? We just don't understand it. It's when neuroscience was founded, by the way, like, one floor away from where I'm sitting right now, right, by David Hubel and Torsten Wiesel, they were interested in vision because as humans, we're visual creatures, right? And so the senses we really understand are vision and hearing, because we talk a lot, we
Right.
communicate verbally, and of course touch. So these are senses, I think, that have been really richly studied by human neuroscience scientists. And smell has been a little bit ignored. And it's been ignored largely because it's thought of as a kind of aesthetic sense, right? It's a little bit of a bonus,
Hmm.
right? Although I would argue that, with COVID, maybe those of you who've lost, folks who've lost their sense of smell, maybe they would gain a greater appreciation for the sense, right?
Hmm.
But really, it's thought of as this kind of thing that isn't that important. But the truth is that in most animals, it's the main sense they use to interact with the world, right? And so, if you're a brain scientist, if ultimately what you really care about is how our brains work and make us who we are, understanding how animals like mice smell and use that information to generate behavior is fundamental. It's not bonus. It's something we really have to understand. But because of just the history of the way that neuroscientists have approached the brain, we know very little. And so at every stage of the olfactory system, there are mysteries. We don't really fully understand how the nose, detects odors. And we certainly don't understand how the brain processes odors to give us the very rich memories that odors evoke, or, how odors are capable of triggering really profound emotions, which is all true, as you know, right?
Mm-hmm.
I'm sure you have smell memories where you walk into a kitchen, reminds you of your grandma, reminds you of being in a particular place at a particular time. And there's something very profound and actually human about that, and that we just don't understand. And so I've been really interested in trying to understand just the basic architecture and function of this system, in part because I think it's just a great way to think about how the brain works. How do we remember things? How do we place sensory cues into their contexts, right? How do we navigate the world? So understanding olfaction is just a really great substrate for just thinking about how the brain works in general. And one of the questions that we've been very interested in asking is, at a molecular level, how does the nose detect smells? Right. Now, as you mentioned, Richard Axel, my postdoctoral advisor, who won the Nobel Prize in 2004 with Linda Buck for discovering what are called the odor receptors, discovered this very large family of genes that encode receptors that are expressed in your nose. And when I started my lab, we hypothesized that maybe there were more and different genes. Maybe this family of genes that Richard and Linda had identified weren't all there was. Maybe there were some that were missed.
Yeah, just to take a little break and to kind of help our kind of more broader audience understand
Sure.
how this would work, right?
Yeah.
So the sense of smell is because of chemicals
Yeah, that's right.
that we sense, that we take in.
Yep.
And the general idea is that in our olfactory system, like
Mhm.
somewhere right behind the nose, uh, there are cells, olfactory cells, that can sense that, and they do that by virtue of these receptors, which are
That's right.
molecules
That's right.
that can bind to one of those chemicals, and they get activated, and that somehow that code creates a certain perception of smell down the road. Like that would be the
That's right.
general idea, right?
That's exactly right. So
And
basically,
what Richard and Linda discovered, one of the seminal discoveries in neuroscience, was that there's a very, very large family of these receptors, right? And that um, they kind of tile our olfactory system to some extent, that allows you to detect, and the combination of receptors that are being active probably creates a sense of a certain kind of smell or complex smell.
exactly.
Um, Now, going from there, you maybe start talking a little bit about in your lab. Um, I guess what is the unanswered element that you're trying to understand? Because like there's a bit of framework. There's lots of like thousands of olfactory genes
That's
that have
right.
been identified. You have this receptor doing it. So what was it that like piqued your interest and to dive into that a bit more?
There's a very basic observation, which is that if you prevent all of those receptors from working, to some extent, you can still smell. And so,
Interesting.
and so there must be some other mechanisms in the nose that allow you to
Right.
do that. I mean, you can't smell everything. You can only smell certain special things.
Got it.
We wondered,
So there was another system
yeah,
in place
that's right.
that detected it. So you guys decided to start looking for it,
That's right.
and through that you guys identified a new class of
We
receptors.
did. We discovered a new class of receptors, a set of molecules that don't look anything like the molecules that Richard and Linda discovered. So they're a completely different way molecularly of detecting odors. And the genes have a kind of awkward name. They're called the MS4As. So I'll just refer to them like that, right? They're the MS4As, and they seem to detect, I think much to my delight, odors that are super important for mice. So things like food and potential mates and predators. So these receptors seem specialized in some way to detect these very special smells that are of particular ethological relevance to the animal.
Okay, so it's a new class of
Yep.
receptors. We'll call them MS4As
Mhm.
because that's the name
Yeah.
they have, and they allow animals to detect signals that maybe the other receptors don't, and of strong behavioral relevance. So are these unique to mice, or do people have these receptors?
People have them. So I think it's one of the interesting things is that these are genes that are expressed in humans. And when we look at data from human noses, we can see evidence for expression of these genes. So it's possible that these genes are acting as olfactory sensors for humans as well, not just for mice.
Got it. Okay. Moving from that and kind of maybe the broader implications of where it takes you in many genes are expressed in several tissues. So the MS4As, are they uniquely expressed in the olfactory system, or are they expressed in other parts of the body or brain?
Right. So that turns out to be a really interesting story. So, the cells in your nose that detect odors are called olfactory sensory neurons, and they're actual neurons, like the neurons in your brain that process information. And it's just they live outside of your brain in the olfactory cleft, the cleft in your nose, right? And so we asked, once we cloned these genes, one of the first things we asked was, well, where else are these guys, right? I mean, and the expectation, the
Hmm.
naive expectation you would have would be, well, if we looked in the brain, maybe we would find different neurons that express these receptors. Maybe they're playing some role in brain function, right? And so we went looking, and that's not what we saw. And we did see that the MS phrase were expressed in other cells, but the cells that were expressed in were cells of the immune system.
Interesting. And this is just a kind of fascinating part of biology and um, evolution, maybe, which is that you think that there's a very specialized cell that is a specialized thing. It has a receptor to do it, like smell, and then you find that the same protein being present in other cells and other
That's right.
parts, right?
That's right.
And often has to have a different function
Right.
because in your case, you're saying when you find them in the immune system, obviously the immune system is not like smelling anything.
That's right.
So it's also present in other cells, right?
That's
So I think that's this I think fact is fascinating, and in some ways then kind of leads you to understand other functions of this family of proteins, right? And I think it gets to
that's right.
maybe carry more recent studies
That's right.
because it also turns out that they are that this class of proteins might also have some association with disease. So how did that happen? So on one side you're studying MS4A in the
That's
olfactory system, you've discovered, and in you're studying sense of smell. From there, how does the story turn to looking at other things it might be doing, and what did you find?
Yeah. So, I mean, that turns out to be, I think, super interesting and surprising to me. So, we started this whole line of research just trying to figure out basic mechanisms through which the nose detects smells. And we stumbled upon this new family of receptors. And we looked through the literature. There was basically nothing known about these molecules. Like, really, we don't know what they do. As I mentioned, we know they're also expressed in the immune system. But there was one other interesting fact that was known about them. And that is, if you have a mutation in these genes, your risk of getting Alzheimer's disease is changed. So depending on the actual mutation you have, that risk can go up or that risk can go down. But it seemed
Hmm.
very, very clear in studies that involve millions of patients from across the globe of every race and geographic region that,
Yeah.
that these genes are somehow fundamentally involved in some
So
way.
let me get a clarification here, because you described as a mutation gene, because I'm trying to map it onto like other risk genes we know about. Like, everyone I think has heard about ApoE as
That's
a
right.
risk locus, ApoE4 as being a risk gene, right?
That's right.
You get your 23andMe profile, and then you kind of gingerly test whether or not you have the A4 allele. Um, and I describe them as variants.
That's right.
So when you're describing them as mutants, I guess I want to distinguish this a
Sure.
little bit from, like, let's say, cancer-causing mutations, which
That's
are
right. That's
de
right.
novo new mutations that happen. So in the Alzheimer's case,
Yeah.
are you talking about like a naturally occurring variant that occurs in a significant part of the population, or an acquired mutation that it confers risk for disease?
Yeah. Here we're talking about very common variants that are
Okay.
pervasive across the population. And actually, if you have one of these variants, the actual amount that your risk is changed for developing Alzheimer's is fairly modest. It goes up or down 10%, 15%. But because so many people have variants in this gene family, it means that in total, the amount of Alzheimer's disease that you can attribute to having these variants is pretty high, actually. So,
Right, got it. So
right,
and just to kind of like talk about the variant concept a little bit,
Yeah.
because I think, when I first started learning genetics, I would think, oh, here's a gene, it has a sequence of nucleotides, and that's what it is. And then over time, I discovered that, well, not exactly. Like for the same gene that you and I both carry, the precise sequence might be a little bit different. Like this just happens in the population. So it's pretty clearly identifiable as that gene, but they're little variants. So my copy of a gene might look a little bit different from my neighbor's copy of the same gene. They're
That's right.
small variants,
That's right.
and sometimes those variants are of a nature that can confer risk, and a lot of risk genetics is around that, is to identify small variations in the gene that are different from other people, that then gives them more risk. And in your case, you're saying then in the MS4A locus, there are these variants that increase your risk of Alzheimer's disease. That
That's
that
right.
is something that was known,
That's right.
and was much known about what this protein does. So
No.
okay,
That's the thing, is that the genes that were linked to Alzheimer's disease, which happen to be some of the genes that we identified as odor receptors, other than that, basically nothing's known about them, these genes at all. There's a kind of they're hard to study, and there's a kind of huge void of information about these genes. And so, we became very interested in just trying to understand like one basic thing. And so the basic thing we want to understand is, um, if you have a mouse and you build a mouse genetically so that it mimics some of what happens during Alzheimer's disease in a human, that mouse will develop all sorts of changes in its cognition and its behavior and in its brain, which you can see and measure and quantify. Um, and those changes reflect kind of weakly what we think happens in humans. And the question is, if we change these MS4A genes, can we reverse some of those toxic changes? It was just a very
right.
simple, very
And I
simple.
think that the things that people are familiar with, uh, I think people know a lot about amyloid and tau pathology as
That's
being
right.
hallmarks of disease, right?
That's right.
And is strongly associated with loss of cognition, etc. It's interesting how our ability to detect these things is like advancing faster than uh, the rate at which we can do something about it, right? So now there's a tau
Yep.
assay,
Yes.
right? There's a blood phospho-tau assay, and I
That's
get
right.
calls from my friends saying, should I, go down to Quest Diagnostics and get a phospho-tau assay? And being the neuroscientist, I should have something to say about it. But it's interesting that we are being able to detect these
That's
changes
right.
at the systemic level, and might allow earlier detection and so on.
Yeah.
And so those are core pathologies for Alzheimer's
That's right.
disease. You have a gene you identify, or that there's an association gene that you're studying, MS4A. It has an association with disease.
That's right.
Tell me a little bit about how does MS4A intersect with these maybe pathologies that we know to be
Right.
pretty common in Alzheimer's?
Exactly. So, we decided just to try to, through a relatively simple experiment, address just that question, right? So we took a mouse that is, in the field is referred to as a tauopathy mouse. So it's a mouse that mimics kind of what happens when you have a bunch of tau in your brain, basically. And in this case, there's an excess of human tau that's kind of in the mouse brain. And that causes all sorts of bad things. And in fact, we did a thing that hadn't been done before, which is to use our behavioral analysis, our motion sequencing analysis, to really very carefully track the ways in which tau changes the behavior of these animals. And it's really quite profound. All of the
Oh,
different
interesting.
elements
Oh,
of
really?
behavior are in the wrong order, or expressed incorrectly. And it's really trivial through the lens of machine learning to distinguish one of these tauopathy mice from like a wild type counterpart. That becomes this very, very easy, to see these
Wow,
differences.
that's actually remarkable,
Yeah,
because I think that although it might seem obvious in the context of this conversation and the platform you developed, but for a long time there has been this challenge, right? Is that you have animals that have a model of pathology, but it's very hard to see any behavioral impact, right?
that
Or reliable behavioral impact,
that's
or
right.
even know is it having a behavior impact.
That's
But
right.
you're telling me that if you make a tauopathy mouse, it has a behavioral impact, and that is detectable
That's
with
right.
precision
That's
in
right.
the platform that you have. Okay, all right. So
That's right. And
so
it
you
just
have a
it's
you have a
and
you have a
add one thing to that to give you a little color. So, one of the complexities in studying behavior in these animals is that often they don't develop their phenotypes until they're old. They're older, right? Like you have to
Right.
wait. Just like with humans, humans don't get Alzheimer's when they're young. Same with the mice; you have to wait until they're a little older. And so there's this
Right.
intersection of age and disease, and you kind of got to tease apart those things in order to really understand the behavioral impacts that are due just to the disease. And so part of what motion sequencing lets us do is actually identify those specific changes in behavior that are just due to age, and to
Right.
segregate them away from those changes that are due to potential disease. And so a thing we can do with motion sequencing is essentially build a behavioral clock. So I can look at a mouse, film it in 3D, and I can tell you with about 2-week accuracy how old that mouse is across its entire lifespan. And so we know a lot about how behavior changes in mice as a function of their age. And we know then how disease might alter that trajectory, over time. And that turns out to be very, very powerful for understanding how disease might, change the ways in which mice interact with the world.
Okay, so tell us now about the intersection between MS4A and tau pathology and the insights that you might be getting.
Yeah. So the first thing was just, we had these tau animals, and their behavior was really changed. If we gave them cognitive tests, their ability to remember was really changed. When we looked into their brains, there were fewer neurons and fewer synapses that connect neurons so that information can be passed between them. All those things were kind of damaged. And when we deleted the MS4A gene, one specific gene called MS4A6D, which is related to the human gene that is changed in Alzheimer's disease, when we deleted that one gene, all of those changes reversed, all of them. And so, no longer was we rescued all of the neurons. So there's a normal number of neurons. There's a normal number of synapses. And these mice, which previously were incapable of remembering anything or even behaving normally at all, they looked pretty great.
So overall, when you delete a gene, often there can be lots of other effects, right?
That's right.
So are you saying that in these mice where you delete MS4A6D, that generally the mice are okay and the pathology is not developing? Something
That's
like
what I'm
that?
saying. That's exactly what
Wow.
I'm saying.
So that is, so what happened? Did you like pop a bottle of champagne at that point? Like what?
No, come on.
You didn't. Wait, you're gonna wait for some fancy paper to be published?
Yes. Well,
Like
not
that
just
is
that.
remarkable. Like that's like an aha moment, right? Like
Yeah.
certainly you would not have expected that. I would
That's
not have
right.
expected that.
That's right. Well, I mean,
I
of
mean,
course,
that's a pretty profound result.
so we're very excited about the fact that, when we mutate this gene, we can reverse a lot of these changes. And we're very excited about that. But the real question for us is why? Like, what's actually happening here to allow these changes to be reversed? And so that's kind of what we spent the last several years focusing on, is not just
Okay,
the
so just
what of it, but the why.
tell me at a very high level,
Sure.
maybe a little bit about what you've learned about how this
Sure.
MS4A6D
Yeah.
gene or the protein is working.
Yeah.
That might contribute to it, like slowing or maybe
Okay, so at
stopping
the highest
pathology.
level, what we think is happening relates to a kind of new view of what's happening during Alzheimer's itself. Okay, so let's just talk about that for a second.
Okay.
So we're used to thinking of Alzheimer's as like purely a brain disease, right? And when you were, a budding molecular biologist, as was I, we thought the whole thing was about neurons dying in the brain and there'd be something wrong with the neurons alone. And that's kind of what was going on, right? Like there were toxic amyloid and toxic tau killed the neurons. That was the story. And what's increasingly true is that many, many different cells contribute to getting Alzheimer's disease. And key amongst those cells are cells in the immune system. So basically, when you get late Alzheimer's disease, your blood-brain barrier opens a little bit, and cells from the peripheral immune system that are responsible for, attacking viruses and bacteria, they actually leak into your brain and are present in your brain and contribute to the damage that's being done during Alzheimer's disease. So your brain becomes really inflamed, basically.
Got it. So immune cells literally go into the brain
Exactly.
and drive disease pathology.
That's exactly. And so we now think of Alzheimer's disease as a neuroinflammatory disease. We think about it as being the consequence not just of tau and amyloid, but also of inflammation in the brain, interactions between the brain and immune cells in your blood. And what we've learned is that this MS4A gene coordinates the ability of peripheral immune cells to get into the brain and to turn your brain into an inflammatory soup. And if you prevent the MS4As from working, then cells from your peripheral immune cell system stay in your body instead of your brain, and that calms your brain down. And you don't get these kind of end effects of having a lot of tau in your brain. It's enough to restore cognition and to save your neurons.
That is super exciting. So do you think that as a therapeutic, then like targeting MS4A is a potential approach to explore? Or are there other reasons why, while this biology is interesting, it doesn't provide a path to potential treatments?
Yeah, we're super excited about this as a potential target for therapy. Yeah. And part of why we're excited about it is, we have data demonstrating that if you knock out MS4As in the brain alone, it's helpful. But also if you knock out the MS4As in the peripheral immune system, it's helpful. And, it's really hard to get drugs into the brain or to knock out genes in the brain, or like manipulating the brain's really hard. But what
Right.
we know a lot about is manipulating the peripheral immune system. We have many, many drugs and many, many approaches to do that. And so it suggests that by giving a drug that just targets your general immune system, a thing we know how to do, we might be able to help with people in the later stages of Alzheimer's disease. So I'm really, really excited about this
Yeah.
as a potential therapeutic approach.
No, I think I love it. I love this story because not only because of the kind of unexpected connections of your basic science research to something that is therapeutically relevant, but it is also a field, frankly, where there are relatively few new targets and few new approaches, right? So if you look at the two approved drugs target amyloid and provide, I would say, somewhat like nominal benefit in cognition, and most of the other approaches just focus on three or four targets. And so having a completely new way of thinking about the disease, I think, could be transformative. And I'd be hard pressed to think of other examples where knocking out a single gene in an animal model has the kind of effects that you're describing. So I very much hope that it finds a way into potential therapeutic and can help people in the future. Given all this exciting work, one of the things that, and I've followed your work for a long time, I was surprised to see a few weeks ago a video that you posted that has
Mhm.
probably been seen pretty widely, and where you talked about something to the effect that your funding for work, including work for Alzheimer's disease, was being terminated. And it was really quite shocking. And of course, it intersected with what is happening in the country right now, generally in terms of funding, but often it is, somewhat general and impersonal. And just to provide context for people who may not be following it closely, in the past few months, the NIH in particular, and in various ways, uh, have taken various moves that significantly reduce funding for biological research generally. In one area, it has been around like the impact on potential indirect costs, and then also in a targeted way. And the targeted way is somewhat relevant to your story. It might intersect in multiple ways, which is that you are at Harvard, and Harvard has been very much in the news in terms of interactions with the government around, at its core, it seemed like the core issue had to do with protests around Palestine and how that was handled and the environment at the university. At least that's, being on the outside, that's the story that we saw. Um, Last year, there were a lot of protests around university campuses around the war in Gaza and Palestine and Israel, and that itself is not surprising, right? I mean, I think that there, uh, maybe for every war that the United States has been involved in, there have been dissenting voices, and it is a mark of democracy, right? So when we were going to Vietnam, there was this question, and those were wars where we were more directly involved. I think historically we've seen it more in direct involvement as to whether we should go to Iraq or not, our involvement in Ukraine nowadays, and then I guess more indirectly here about something that's happening in Israel and Gaza. So that drew a lot of attention. And at some point, we heard that like part of the way Harvard was addressing it, and I really don't know the nuances of it, so we'll not go into that detail, but one of the consequences was going to be to cut significant amount of funding or freeze funding that was going to Harvard, right? And so you see it from a distance, and for me, what was surprising was, I guess, is potential lack of connection, right? So I think the topic of what free speech looks like, what the boundaries of free speech are, how do you create an inclusive environment um, so that all voices are heard. I think those are all important points, and they should be discussed. And if they're not being addressed, I think one should call it out. But I was surprised that the result of that was to kind of freeze a lot of research funding that had been granted based on high quality science in across many disciplines. And even though it was fairly theoretical, I said, like, okay, I'm not sure I agree with that particular avenue. And then, and you posted this little video clip um, that crossed my feed, and I said, wow, this is quite real. And in a lab whose work I followed, and a person who I know is doing really important work. And so I'm sure that is not the intention. Like I would say that, whoever's sitting in government trying to kind of improve the system does not really intend valuable work to be slowed or stopped, right? So tell me a little bit about that, because I don't know any of the backstory. That's just what I saw.
Mhm.
And we've talked a little bit about your research, but how was the funding for this research affected? And then I guess what prompted you to start speaking about it?
Right. So, I mean, the first thing I'd like to say about this is that um, the research that we do here at Harvard, and really the researchers across the globe do, focused on um, diseases that are just critical to us as a human race, things like Alzheimer's disease, Parkinson's disease, is in many cases supported by public funding for research. And part of why America is so great is because for 70 years there's been a sustained, consistent investment by the public in improving our understanding of basic science and disease science, and that has yielded not just unprecedented economic growth, but a huge amount of knowledge that has been used, for example, to take the death rates from childhood leukemia from 90% in the early 1970s to almost 10% today, right? We've basically cured this disease, this devastating disease, through science. And that really has been the product of sustained public investments, especially here in the United States. And I'll say that there are many, many things about the system that could be improved in many ways, for sure. Like if you've been in the system, I've been in the system. We know the system's not perfect, but think about the greatness of a system that allows someone like me, who's never studied Alzheimer's disease, to get a grant from the National Institutes of Health to explore this important possibility, right? That maybe this particular gene could be helpful, right? I mean, think about like the trust that the reviewers of these grants must have had. Think about the structures that must have been built in order to allow such a grant to be issued, and the public faith in science to want to invest in that way. So to someone like me, I've always been super grateful and proud, to be honest, of like what we've collectively accomplished in this country over the last 70 years of continuous funding. And I couldn't be more grateful at the opportunities that the public has afforded me, honestly, right? I feel like what I do is public service. That's honestly how I feel. And to be as direct about it as possible, I know that that's why most of the people in my lab do what they do. They're grateful for the opportunity to do science. They feel like it's special and rare, and a sign of an advanced society that's invested in its own people, and they just want to give back. They just want to give back. They work the 60 hours a week, not because they want to work harder because they're ambitious. They do it because they're deeply curious. They want to understand how things work, and they want to give back to the people who've supported them. And so, like, all of my feelings about what's going on now start from this perspective of gratitude. Just to be clear, um, earlier this year, as things developed with Harvard and the Trump administration, basically, many of us were a little bit watching from the sidelines. We're hearing things in the news just like you, and we know that there's some sort of rift between the Trump administration and Harvard over many issues. And then essentially, without warning, all of my grants and all of the grants of my colleagues got terminated across essentially all areas. So not
So you
just,
just got an email or a letter saying that
yeah,
your grants are now terminated?
actually, in some cases, not even an email or a letter, just the system that administers grants indicated that the new end date of the grant was tomorrow. And and that's it. So with almost no explanation whatsoever. So just essentially a summary termination, independent of the topic of the grant or the people doing the work or their stage of training, independent of any of those things. Just the wholesale deletion of all federal funding. So for me, that included a grant that I had with NASA to do this kind of behavioral analysis in space, which would help us understand how behaviors change as you go to Mars. It affected all of my grants related to the sense of smell, which is of course still important, given that there's a couple million of people who can't smell because of COVID, right? And it included our work on Alzheimer's disease. All of that funding, basically in a matter of 2 days, was eliminated, and all the support for the trainees in my lab went with it.
Right. I think, and there's a component of the how here. It sounds to me like, the grant administrating system is pretty uh, sophisticated and evolved. They're program managers for all the grants.
Mhm.
But you're saying that it was not the case that somebody called you and said, I want to talk to you, Dr. Datta, about what's happening. And for these reasons, we need to stop or pause the grant, or what the context is. And I guess also some thought about the impact of it, right? Because in your labs, there are postdocs and students who are working. So it's not just the animal experiment that is affected. Everybody who is working on that is affected as well, right? And so it has a lot of these secondary consequences. It's not
That's
just like, okay, it affects the research, but the students and your postdocs who are doing the work are affected um, in in a in a way that seems kind of like almost immediate, right? So
that's very
so you just find out all of a sudden without much context. I can't quite imagine what that situation would look or feel like, but like, what was your reaction? Like, you described it in a very factual way,
yeah, that's right.
but
That's right.
I mean, I don't know what I would be doing. I don't
Yeah.
know if I'd be pounding my desk or cussing or trying to call people, or I don't know if there's a 911 for grants. So
Yeah,
like, what was your reaction? Like it's like, even knowing the broader ecosystem and what is happening in these questions, it must be a shock to the system just to find out, there you
that's right.
go, your grants are stopped, right?
That's
So
right.
how
Yeah. I
how
mean,
did it feel? How did you deal with it? Or
yeah, I mean, I was totally stunned. I never imagined that this would be a thing, right? I mean, I think none of us went into science thinking that one day, essentially out of the blue, we would just be told to stop. And I will say the one thing I will say about this is that the day that it happened, I vowed not to stop. That was like the one reaction I had, is that no matter what, I'm going to do whatever I can to keep going and to support my people and to help them keep going, because what we're doing here is important and we believe in it, right? I believe in this process. And so, as best we can, we're going to try to keep going. That being said, it's hard to do research without research funding, right?
right, yeah. Yeah,
Right?
absolutely. So I think that, I like your immediate reaction, better than, running around the lab screaming and just freaking people out.
Yeah.
But I would say after that,
Right.
when I started hearing about this, I would again hear it in aggregate numbers. You read a
Sure.
newspaper, it says a lot of grants have been cut. This is the amount of money, and you have very large sums where you don't know
Yeah.
what it means at individual lab levels. And then when you would talk to some of my former colleagues or people I knew at the universities, and this happened across the board. Like, Harvard was particularly hard hit, but there are similar kinds of
That's right.
grant cancellations in many places. Frankly, without much clarity as to how or why it was happening. I think that I would say one of the things that is particularly concerning is not just that a particular grant gets canceled and there might be reason that it's no longer relevant or something like that, but we work in a system that works a certain way, right? And the United States is one of those things. And having lived in, and worked in many parts of the world, I know it is unique and singular in it being a non-corrupt system, with transparency. Even the grant review process, it's a masked grant review process. You don't know who reviews your grants, um, and and then you rely on the system. Whatever, it's like we rely on our police to be there if
That's
if
right.
if there's a crime. We rely on our hospitals and doctors to be there if I need medical attention. I just
That's right.
assume that those things would be there.
That's
And if
right.
all of a sudden tomorrow it says, the hospitals are shutting down, it would be a huge hit to the system, right? And so I think there's this element that it seems like a system that we believed to be immutable or to be a pillar of the foundation of the way work is done in this country was at risk. Uh, Having said all of that, and many people feeling that, when I would talk to people, they would be reluctant to come forward and talk about how it was affecting them. And I could not tell what it was. I could not tell if it was shame. I could not tell if it was fear. I could not tell if they didn't want to be in the limelight and anything else. But I thought it was important to talk about it because I said, other than that, it's just some vague number. Harvard has a lot of money. They'll probably figure out how to solve the problem. And maybe it is not having much impact. Maybe it is work that's not valuable. And especially if you're not in a university setting, or biotech, it's a pretty opaque system, right? You
Right.
don't know how it works and you don't know what
Right.
the impact is. So I thought it is important for people to come and talk. You made the decision to speak about this, right? So tell me a little bit about that. So from the event to your decision to speak about it, why did you think it was important to talk about it? And maybe what were you hoping to achieve? And like, what has the reaction been?
Yeah. I think the thing that you're noting, which is that many people are scared, is very, very true. So I see it in my own trainees. We see it across the university and nationwide. I mean, people are terrified, and there are real consequences of these funding cuts to people's lives right now. So, many of us, for example, have students who are deciding to leave science, which is like an incredibly depressing thing. Students who've trained with us for 3 or 4 or 5 years, postdoctoral fellows who have PhDs and are talented and dedicated. The uncertainty and the anxiety that all of this has engendered has changed the way they view science in this country. And I think we're going to be feeling those effects, for many years to come. And my sense is that in this time when so many of us are afraid, it's especially important just to speak out. It's not about politics necessarily, but just about what's happening. Just
About impact. No, I agree. I mean, I think that in informed citizenry is very important, right?
yeah.
And so, having said that, I think the choice is not an easy one. So I do want to ask you a little bit about that, because I think that there might be a lot of people like you in different places who are affected and who are wondering, like, they're having a conversation at home and their spouse is saying, I don't know if you want to get in the limelight. I don't know, it has little upside and maybe some risk. I, like you, believe that it's very important to talk about these things, and
Yeah.
and I think that, um, frankly, the freedom to express yourself, the freedom of speech is our most important attribute as a country, and so I think that um, we should use it and be heard. Having said that, when you were making the decision to come out and talk about it, was that was that another thing that you said? Just like you said, I'm going to continue the research. You just said I have to go out and talk about it, and I'm not going to seek permission or get some talking points from the PR department at Harvard or whoever else. But I'm going to express myself.
Yeah. I feel really strongly about this. I mean, I think you can tell that, I feel like this system of, government supported research is part of what's made this country great and has made me so proud to be an American. Honestly, Anirvan, I'm a patriot. I believe in this country and in this system. And, there are real consequences to these cuts. And I think people are unaware of them. And so for someone in my chair, I feel like I've had so much privilege. It's imperative that I at least describe what's happening. And if that does any good, I'm grateful for that.
Right, and I think that I think there's an opportunity to also engage productively to address what is happening. Right, so I think that to some extent, you face the consequences of whatever happens at a given moment. But then there's a question about, then you decide how you react. And frankly, that's probably a whole entirely different, interesting behavioral study. I've often thought about this thing, because there is this moment between a circumstance and your reaction. And maybe there's a name for that moment. I don't know. I'm not a behavioralist. Uh, where the brain is processing about, how am I going to react to this situation, right?
Yeah.
And part of it is just programmed. It's just the way your brain is wired. This happens and you do this. And then part of it is a thoughtful reaction, right? And there's an opportunity, I think, for all of us to take that moment and really think deeply about how we're going to react. I mean, frankly, the creation of this podcast
Mhm.
is partly a reaction to the circumstances. So I saw many aspects of our society being affected by uh, various decisions that were being made. And I felt that at least one element of what might have contributed to this is a weirdly fragmented society, that we did not know what other people do, what
Yeah.
is important, what is not. Maybe that leads to some level of lack of empathy for something that's going on. And you just end up believing whatever you're hearing in your small domain, right?
No.
You're an academic, you talk to other academics, those are all your friends, they come, and you have a somewhat similar political leaning or worldview, and you talk, and maybe you complain about the way things are. But nobody else gets to understand why you're upset or why it matters. And likewise somewhere else. Like, I feel that it's a vast country. And there are likewise many things that I think we don't pay enough attention to, just because we don't know, right? I mean, I think if you I've seen it, like, driving through rural America, I'm always struck by, and I feel worried and sad about when I see communities that feel like they're not thriving. You just go, you can see, you see the poverty. And I remember I was like driving through some area, and I was telling my wife about, a bit of a stark contrast with what, like, let's say what villages in Switzerland look like, where I'd spent some time. That there's a little thriving community in the middle. All the houses, the farms around the circle, there's a little community center, there's heart. It is viable and sustainable. Whereas many small communities in our country are not. And oftentimes we don't know that. It doesn't make the news. Often we don't know what it means for the people and how to help them, right? So there's that. And likewise, for other domains, there is a misperception around what the world looks like and what people are really doing, right? I mean, I think that so I felt that a podcast would be a way to somehow broaden the conversation, right? Just for people to know, just like I think we should know what is happening in rural America, what is happening in small towns. We should know what is happening in poor communities. We should knowing what happened in technology. And way too often we get stuck in our little bubble and we don't take the time to go out and speak, right?
Yeah.
You spoke very eloquently about the discovery of the receptors that you had worked on, the implication of Alzheimer's disease. But face it, most of the time for the last 10 years, you would have given seminars. You would have gone to other places and
That's
conferences, right?
Yes.
And scientists would have understood it.
Yes.
And we just did not create a system to acknowledge that fact. Like, it would show up in a slide that the work is funded by the NIH or by Howard Hughes or somebody is funding it. But I feel that we probably also have an obligation, maybe collectively, maybe that's what we start doing. Like, if you don't want to speak about what is happening now, just go and talk about what you're doing and why it is important, right? Because people are funding that work.
Yeah.
And they deserve to know, right? And so it can be explained often in ways that people can understand. It can be done in colleges and communities. I greatly appreciate the time that you have taken today to talk to us about the work in your lab. I think it's very important, and I'm very excited about the intersection with Alzheimer's, just being a neuroscientist and a drug developer. But I'm also very grateful for your willingness to come and speak about these things candidly. And I feel that the country is in a lot of need for courage in every corner, and your way might be different. You just have to find, how do I show up and talk about what my experience is, right? And so for somebody, it might be to go and protest. For somebody else, it might be to just explain why it is important, even from the government side equally, right? If you're investing in a certain thing, maybe it is important for us to understand a particular foreign policy element, right? Just instead of saying this is what we're doing. And one of the challenges is we live in a world where there's so much hyperbole. Everything is great and super. It becomes difficult to know what to pay attention to, right? If everything is loud and making a lot of noise.
That's right.
But in my mind, I think what is happening in the scientific establishment, the potential risks, not only at the level of individual labs like yours, but more systemically about our review processes, the way the FDA is organized or the CDC is organized. We implicitly really rely on a system that works well, that we trust, that we believe that if the government approves a drug, that it is a safe drug that is going to help us. And if you start losing faith in that, then very quickly you're back, I don't know, 300-400 years. So I think it's really important to kind of go out and explain why it's important and do what we can, and actively as individuals, and not just say, well, somebody else is going to do this. Like, I think this is the moment to really say, what do I do? And I would just like maybe close by asking you, like, you of course have talked a lot about what you're doing, but I would love to hear your thoughts on both about what you think you can be doing more in this moment, but also maybe the broader scientific community and what's important for us to do collectively as a society, so that we end up with the right solutions.
So I think a thing that's important to recognize here is that all of us have more in common than not. Right. So it doesn't matter whether you're in a blue state or a red state, whether you're rich or poor. If your parent gets Alzheimer's disease, there's still very little we can do. Doesn't matter. right? Like the outcomes aren't that different. And for lots of problems related to human health, this is true, right? We're all affected by our lack of complete scientific knowledge and our inability to tackle some of the biggest problems that affect our collective health. And I think no one, regardless of party or geography, thinks that the way we're going to ultimately deal with these problems is through less innovation or less science or less research, right? I don't think anyone believes that. I do think that people might have a misunderstanding of what it means to do research and the ways in which research can impact your lives. And so I think that trying to meet people where they are and emphasizing the fact that we all have this in common, which is so true. Like we all live on the same planet with the same set of facts, basically.
Yeah.
And right, like in the end, that's true. And we all face at some level very similar problems, and we can help each other solve them. Right.
And I think, I feel that, this ability or frankly developing the ability to talk to people who might hold different views on you on a certain topic, but talk productively about an important issue is super important.
Yeah.
And I say this because I feel that we tend to paint other people in broad strokes and sometimes unfairly. And I'll since you talked about, I'll talk about the political affiliation thing a little bit, right? So there is a segment of society that will think, or maybe among Democrats or blue states, that the Republicans must be against science, right? I think that is absolutely not true. I know it is absolutely not true, right? I know people, I've known people throughout my life from both political affinities who have held similar views on a wide range of topics, right? And so, and likewise for any other topic. And I think it can be very frustrating. You can imagine, right? If somehow what the administration is doing is somehow used to broadly paint a certain sector of society as not caring about science, not understanding it, it is absolutely not true. And so I think we would all do well to take some time, make an effort to talk to people who might not hold all of our views on society or how it should be organized and the financial system and all of those things, and to be able to talk about things that are important in innovation. Like one of the things that I like a lot about being in biotech, frankly, it's a very mixed environment.
Yeah.
And it is politically mixed, it is economically mixed, and you're always working with people who hold lots of different views, but it doesn't at all get in the way of deeply understanding, well, is this a good idea to invest in as a biotech company?
That's right.
Is this something that we should be doing? The same thing, should we go to Mars? So I feel that those things that I think collectively as a country we're very proud of completely cross political ideology. And I think we really need to somehow not make that the center of the story.
Yeah.
Right. And I appreciate everything that you're doing towards it. And I hope everyone who's listening to it can do their part in making it happen. Well, thank you, Bob, for joining today's call. I really enjoyed our conversation, and very much look forward to seeing the evolution of your work and how it can impact what we're doing.
Thanks very much for having me on, Anirvan. I've really enjoyed talking about the science, but also about the implications of what's going on for society. And I think I very much agree with you. The more that we can just talk to each other, in a way that allows us to emphasize our shared values and to recognize that we're all kind of in the same country together, the better off we're going to be.
Fully agree. All right. Thanks, Bob. Take care. I'll talk to you later. That's it for this episode of Let's Talk About That. Thanks for listening and for being part of these conversations that explore the ideas, the people, and the events that are shaping our world. If you enjoyed the episode, please share it with your friends and family and take a moment to rate and review the show wherever you get your podcasts. It helps others find us and keeps the conversation going. I am your host, Anirvan Ghosh. Our editing producer is Joel Slabaugh, who works his magic on the raw recording with all of its flaws to create a listening experience worthy of your attention. Thanks again, and see you next time.